Hello everyone, welcome back to day 2 of DNA history and replication. So we are going to pick up right where we left off. So we have been talking about part 1. So how did scientists learn that DNA is the genetic material of life? And thus far we have talked about three groups of scientists. We've talked about Griffith, Avery McCarthy McLeod, and Hershey and Chase. So we are going to pick up right where we left off. And we are going to now think about, well, now what, right? We just figured out that DNA is the genetic material of life. So moving forward in history, what were scientists interested in next? And so you can imagine that we knew what DNA was made up of. So we knew it was made up of nucleotides. We knew what those were composed of. We knew it was the genetic material of life. Well, now people were really interested in the structure. So what did it actually look like? How did those nucleotides come together? You and I know that DNA is double-stranded, but that was not known for a long time. So I encourage you to start with a list of things that you remember about DNA. It has been quite some time since we have talked about nucleic acids, particularly DNA. So go ahead and pause the video, and then when you come back, see if your list matches my list. So I'm going to just jot down a couple of different things here. And the first thing is that DNA is double-stranded. Usually, right? So in the case of viruses, we just talked about in day 1 how viruses can have single-stranded DNA, but in living things, DNA is double-stranded. And if you recall, we said that the monomer is a nucleotide, and a nucleotide is made up of a sugar, a phosphate group, and a base. So we can go ahead and kind of draw a little picture. So here is our sugar and a base attached to the sugar and a phosphate group. And we can have 4 bases, A, T, C, and G. And our polymer is called a nucleic acid. And then the bond, let me know if you remembered the bond name. The bond name is called specifically a phosphodiester bond. And for our base pairs, A pairs with T and C pairs with G. And then last but not least, we can also remember our 5' and 3'. And DNA is anti-parallel and it is complementary. And that idea of complementary goes back to A pairing with T, T pairing with A, and then C's and G's pairing together. So hopefully your list matches up my list. And let's go ahead and start with our first scientist of today, and that is Chargaff. And this is in 1949. So for him, his question was a little different than the questions that we've been working on answering thus far. And his question was, what is the base composition of nucleotides in organisms? So we knew that nucleic acids were made up of A's, T's, C's, and G's. So we knew what DNA was made up of, but he was really interested in, well, how many A's, how many T's, how many C's, and how many G's. So for his experimental setup, so what he did was he separated DNA from the other macromolecules, and then he treated with acid. And the reason that he treated the DNA with acid was this is going to break those phosphodiester backbones. And then he separated the bases and then analyzed using something known as UV spectroscopy. So let's talk now about his outcomes. And one more thing, actually, before we talk about his outcomes, one more thing. When he did his experimental setup, so this idea of breaking open cells, taking their DNA, treating it with acid to break those phosphodiester bonds, separating the bases and analyzing them, he did this for many species. So his hypothesis was that if DNA is the genetic material of life, that you should see different amounts of A's, T's, C's, and G's in different species, because the order of A's, T's, C's, and G's must mean something, and that must somehow be how this message is translated. So indeed, that is what he saw. So for his outcomes, what he saw was that the base composition varied between species. And again, this is consistent with the idea that DNA is the genetic material of life. And so using his data, what he did was he came up with something that is known as Chargaff's rules. And for his rules, his first rule states that the percent of A equals percent of T, and percent of C's equals percent of G's. His second rule was that the percent of purines equals the percent of pyrimidines. So recall from way back when in the semester, for purines, purines are pure as gold. So a purine is A and G, which makes our pyrimidines T and C. And pyrimidines have one ring, purines have two rings. And so for the percent of purines equals percent of pyrimidines, he saw that there was always 50% of each. So regardless of the species he looked at, there were 50% purines and 50% pyrimidines. And so keep in mind, these rules, this was before we even knew what DNA looked like. And these rules were really important for Watson and Crick later using some of them to solve the structure of DNA. So let's go ahead and using these rules, let's do a practice problem. So pretend that you have isolated an organism on campus and you find that its genome is 30% A's. Based on this, what percentage G does its genome have? So go ahead and pause the video and work through this practice problem and see if you got the same answer that I did. So if we have 30% A, from Chargaff's rules, we know that the percentage of A equals the percentage of T. So this means that we also have 30% T. So if we take 30% and we add 30% to that, that puts us at 60%. 100 minus 60 is 40%. So that means that the 40% of the genome that is not A's and T's must be C's and G's. And we can take that 40% divided by 2, since we have 2 nucleotides left, the C's and the G's. And that tells us we have 20% C and 20% G. So the answer to this practice problem is B, 20%. So let me know if you got that right, and let me know if you need help with problems like these. So we're going to jump forward a little bit here. And the reason that we're going to jump forward is we're going to remind ourselves of what DNA models look like, and then how this ties back to Chargaff's rules. So on this page here, we have different models of DNA, and they all mean and they all show the same thing, just with different representations. So all the way on the right, we've got a string here of DNA with the A's and T's shown as rungs of a ladder. Here we've got the ball and sphere model of DNA. Here we have more of our cartoon image. Here specifically, we're looking at just the bases, so lots of different ways to represent the same molecule. Now, let's go ahead and take a look at this for a second. We said that one of the rules is that A's and T's always pair together. So here is an A and a T paired together. You'll see that there are 2 hydrogen bonds, and a T has 1 ring, it is a pyrimidine, and an A is a purine. So we have a purine that is hydrogen bonded to a pyrimidine. And same thing for G and C, except in G and C, we have 3 hydrogen bonds. And this image here does actually a really nice job of showing us that it doesn't matter which side the A or the T are on, they're going to have 2 hydrogen bonds. Same thing for the G and C. And so when we think about Chargaff's rules, those rules really make sense when we look at the structure. We always have G hydrogen bound with C because of the molecular makeup of G's and C's. We always have A's bound with T, again, because of the molecular makeup. And we're always going to have 50% purines and 50% pyrimidines because the purine must hydrogen bond with a pyrimidine and vice versa. So it's kind of amazing that he was able to figure this out before we even knew about the structure. So that takes us to the question of, well, how did we learn about the structure? So to first introduce how we learned about the structure, we have to talk about Rosalind Franklin. And she did a lot of this work in 1952. And her question was, what is the structure of DNA? So the way that she determined this and her experiment setup was using something known as X-ray diffraction. And this method is something that is still used, and in the field of virology, we still use this. And I am not a crystallographer. I do not understand how to read X-ray diffraction patterns. It's amazing to me that people do. This is something that you have to have a specific set of skills to be able to do. And Rosalind Franklin most definitely did. So let's go ahead and draw a rough sketch of kind of how this works. So she worked with an X-ray source, and she had her sample, and in her case, that sample was DNA. You can also do this for proteins. We've done this for other nucleic acids as well, so for RNA. And we're going to have beams of X-rays that are shot at that sample. Now, that sample is going to diffract the X-rays, and at the very end, what we had was a film, and those X-rays were then able to shine onto the film and create a pattern. And the pattern that she saw, I'm not doing a great job of representing it here, but is probably one of the most famous photos, and that is photo 51. And to you and I, this probably doesn't mean much, but to her, she was able to exactly interpret what this meant. And so when she saw photo 51, and when she saw that diffraction pattern, what she learned was 2 things. She learned, number 1, that DNA is helical, and number 2, that the bases have regular spacing. Unfortunately, before the Nobel Prize was awarded for the discovery of the structure of DNA, Rosalind Franklin actually died of ovarian cancer, and it's thought that it's likely due to her work. So it's thought that she was exposed to a lot of X-rays because much of her work dealt with using X-ray diffraction and making X-ray diffractions and solving the structures of many things. So it's thought that she passed away due to that. So we're going to come back to Rosalind Franklin here because we cannot talk about Watson and Crick without talking about Rosalind Franklin. So let's go ahead and introduce the scientists of Watson and Crick. And in 1953, these papers came out, or this paper, I should say. So for Watson and Crick, they were interesting fellows for sure. And so they published the paper of the molecular structure of nucleic acids, and it was pretty customary at the time to have very different writing than what we see now in scientific writing. So in this red box, that I've circled here, it says, we have recently proposed a structure for the salt of deoxyribonucleic acid, which, if correct, immediately suggests a mechanism for its self-replication. So they were kind of cocky, to say the least. Not only did they, steal work, which we'll talk about here in a second, but they also went ahead and said, oh, well, we know the structure, so obviously we know what the mechanism is as well. So we'll actually refer back to this mechanism here when we get to the mechanism of DNA. But let's talk about how they solved and what their question was. So their question was the same as Rosalind Franklin's, and their question was, what is the structure of DNA? And how they solved it was they built a model. And they built the model based on a couple of things. They built it based on Chargaff's rules and Franklin's data. So the model that I'm talking about is the model in the picture here on the left. So you can see made up of paper here and aluminum as well. They actually went through and cut out all the bases and started trying to play around with them to figure out how these pieces went together. So you and I know that the bases face inside. At one point, they had a model where the bases faced outside, and there's a lot of different ways to tell this story, and I probably will not do it justice, but I'm going to do my best. So at the time in 1952, 1953, there were a couple groups that were working on trying to determine the structure of DNA. So Rosalind Franklin was part of Wilkson's group, and so Wilkson was her principal investigator, the person that she worked for, and at one point, Watson and Crick were also working on the structure of DNA. It was kind of like a mad race to figure out, to determine who was going to figure out the structure first, essentially. And so Watson and Crick took kind of more the hands-on approach in terms of trying to use bond angles and things like that to build the molecular structures of the nucleotides and then try to piece things together. And they were kind of stuck with how these pieces came together until they actually were able to see Franklin's data. Now, Franklin's data was shared without her knowing that it was shared, and it was shared by Wilkson. There's some thought that it was shared at a bar at one point, because the story goes that Watson and Crick walked into a bar, and one of them had announced that they had figured out the secret to life, even though they didn't have all the pieces quite yet. But so anyways, at one point, Franklin actually saw Watson and Crick's model, and she made the comment of, oh, well, that's a pretty model, but can they actually prove it? So there was definitely some tension between Franklin and her boss, and then her boss was actually friends with Watson and Crick, which is likely part of the reason why he shared her data. And so using Franklin's data, they actually were able to figure out, helical equals spacing between the bases, and they were able to, before Franklin and her boss Wilkson published their paper on the X-ray diffraction, they were able to publish their paper. And it actually came out in the same issue, and that was kind of worked out between them. But kind of moving forward to 1962, Watson, Crick, and Wilson won the Nobel Prize, and Franklin's name was left off of it. And part of the reason there is because she had passed away at the time. But, there's still arguments over if she was alive, would her name have been included on the Nobel Prize? But anyways, definitely in science, there is drama. There's definitely people who sometimes work on collaborations and then people who try to scoop other people for data. So not entirely surprising. Things like this, unfortunately, still happen today. So kind of moving forward now, let's go ahead and jot down what our conclusions are so far. So far, all of our experiments that we have talked about have shown us that DNA is the genetic material of life. And our second major conclusion, based on the combination of Chargaff's rules and then Rosalind Franklin, Watson, and Crick, is that DNA is double-stranded. It is helical, it's anti-parallel, and A pairs with T and C pairs with G. All right, so kind of moving forward, there are a couple of key things that we have to remember, and this is going to help us with understanding how DNA is copied. And even when we move on into the next topic where we talk about transcription and translation, we're going to be seeing a lot of 5' and 3' ends, so it's important for us to mind our 5' and 3' ends. So for DNA, you've already seen me draw this, but again, repetition is the key to mastery. So when we have DNA, DNA is anti-parallel and it is complementary. And one other thing that we're going to summarize is when we saw A pairing with T, there were 2 hydrogen bonds. And C paired with G, we have 3 hydrogen bonds. And this will become really important as we start to think about the molecular mechanism of how DNA is replicated. So for now, we're going to kind of put a pin in that, and we're going to recall these things here in a little bit. So now we are ready to move on into part 2. So how the heck do we copy DNA? So I've inserted one of my favorite YouTube videos listed below here. So I'll also link it in the description box, and I highly recommend that you watch it. It's really, really cool. So let me know if you do watch it, and let me know if you enjoy it as much as I do. All right. So kind of getting started with how the heck do we copy DNA? There's, as you can imagine, lots of different potential ways. So what I want you to do in the comments below is tell me, if you were designing DNA and you were designing it, how would you make it so that organisms copied DNA? And we're going to pick up and kind of reiterate this quote here from Watson and Crick. So this was in their paper from Nature in 1953. So it was in that red box that I showed you. And Watson and Crick made the statement that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So of course they proposed a model, and so we want to know, well, how do we know if the model is right? So we are going to use the scientific method. So what we're going to do is we are going to come up with possibilities. We're going to come up with a hypothesis, and then we're going to test and analyze the results. And this is exactly what scientists did in the day. One of the models that Watson and Crick came up with was a possible model, and their hypothesis was that DNA was replicated in something known as the semi-conservative manner, but there were also other possibilities and other models. So let's go ahead and draw what those possible models were. So what we're going to do is we're going to color code things. So black will be old DNA, and then we're going to do pink as new DNA. And we have 3 models. We had the conservative model, we had the semi-conservative, and then we had something known as the dispersive model. So for the conservative model, the hypothesis was that we started with our old DNA. That DNA was split into 2. It was copied, and then it was rearranged so that the daughter strands were all new. So we had, at the end of a replication cycle, all old DNA and then all new DNA. So the reason this model was called conservative was because it conserved the original DNA that we started with. Now for the semi-conservative model, the hypothesis was that the original strands of DNA were separated and then they were copied, and at the end we had half old and half new. And so this is what Watson and Crick hypothesized. So at the end of their paper, where they suggested they had come up, or they had suggested that the structure of DNA lends itself to a mechanism, they hypothesized that this was the replication mechanism. Now, the third model, which was the dispersive model, was kind of wonky. For this model, it was hypothesized that there wasn't really rhyme or reason. Some parts were old, some parts were new. So you got a mixture of some old and some new. And the idea was that it was a random mix of old and new. So now we have come up with our possibilities. The hypothesis was based on the structure that semi-conservative seems to be the model that maybe is how DNA is replicated. And so now we have to test it. And the scientists that tested the possible models for DNA replication were known as Meselson and Stahl. So their question was, how is DNA replicated? And for their experimental setup, they actually used radioisotopes. So the radioisotope that they used was N15. So what they did was they labeled the nitrogenous bases in DNA, and they worked with E. coli. So specifically, they labeled the DNA of E. coli, and then they followed up with something known as cesium chloride gradients. And cesium chloride gradients separate by density. So let's go ahead and talk a little bit about how the heck did they radio label, what the heck is a cesium chloride, and what does that even mean? So we're going to draw some more flasks. So one of the experiments, or one of the things that they did was they grew E. coli in media, and the media that they grew E. coli in had N15 added to it. So what that meant was when they grew that E. coli, it used the N15 in the food of the media, and it incorporated that when it made its bases, which meant that its bases had N15 in the nitrogenous. So the nitrogenous bases had N15, and then those were then incorporated into DNA. So they could also, instead of growing in N15, they could also grow in N14. So we'll make N14 pink, and then we'll keep N15 black. So they grew E. coli in media, and they could grow it either in N15 or N14. And then they lysed the bacteria, and they took the DNA. So they were able to separate out the DNA from all of the other components. And then they followed that up with something known as a cesium chloride gradient. And this is a form of centrifugation. And what cesium chloride gradients do is they separate by density. So the more dense something is, the further down in the tube it will be. So what I'm showing you here are two things. Here all the way on the right, this is actually a cesium chloride gradient that I did in graduate school, and you can use cesium chloride gradients to separate not only DNA, but you can use it to separate all the components of cells, but you can also use it to concentrate and separate viruses. So this blue band that you see down here, these are my viruses, and what you see up here, this cloudy stuff, this is all of the other cell components. So these are some membranes that are left over from the cell. So because the viruses down here are further in the tube, they're actually more dense than the pieces are up here of the cells. So we can take that same principle and apply it to this idea of N14 and N15 DNA. So if we have bands on the bottom, these are going to be bands where we have N15/N15. So what I mean by that is both strands of DNA have N15 in their nitrogenous bases, and we call this heavy. And we call it heavy because it goes all the way to the bottom of the tube. We can also get DNA closer to the top, And this is going to be N14 DNA, and we call this light. Now, if you get something in the middle, so if you get a middle band, this is going to be half N14 and half N15. So you can think of it as the middle or the median, if you will. And so this last image here on the right shows the same thing that we just drew out. So Meselson and Stahl used these ideas in their experiments. So let's go ahead and walk through step by step how they did this. So what we are going to do is we're going to draw their flask or their media. We're going to draw their cesium chloride gradient tube, or that centrifuge tube. What did that look like? And then we're going to draw what the DNA looked like. So in their first step or their first flask, what they did was they grew E. coli in N15 media. So let's draw some cells in here. So if they grew E. coli in N15 media, they took a sample of that, they lysed the cells, they took the DNA out, and then they did a cesium chloride gradient, which is again just a type of centrifugation. They saw one band. And so they saw an N15 band, or a heavy band, because all of the DNA was labeled with N15. So let's go ahead and draw a DNA strand here. So we're going to make black our N15, and we'll keep our colors consistent, and then do pink for N14. So then what they did, after they made sure that all of their E. coli DNA was labeled with N15, they then transferred some of those bacteria to media that was N14. All right, so when they transferred the E. coli to N14 media, and then they took a sample of that, they lysed the cells, and they took the DNA and put that onto a cesium chloride gradient and ran the cesium chloride gradient, they saw one middle band. And for the middle band, we said that if we get anything in the middle, it's because it's a mix of N15 and N14. So if we go back to our models that we have here, we can cross a model off at this step. So if we take a look at the conservative model at the end, we have all old, all new. If the conservative model were the model for DNA replication, during this step, Meselson and Stahl would have seen two bands, a light band and a heavy band. So we can actually go ahead and cross off the conservative model. And so that leaves us with either the semi-conservative model, which is half old, half new, or the dispersive model, which is part old, part new. Both of these would have had one band in the middle of that cesium chloride gradient. So at this point, they were able to cross off one of the models. So they went ahead and they transferred again. So they took another flask, and again, they transferred. And when they transferred, they were still in N14 media. And just like in the other steps, they went ahead and took a sample of the cells, they lysed them, they took the DNA, they ran it on a cesium chloride gradient, and what they saw on their cesium chloride gradient was that middle band, and we said that that middle band is N14/N15, and they also saw a light band, which we said that the light band is N14. So now at this point, if we go back again to our model here, we can actually start to cross off another model. For the dispersive model, if you always get random mix of old and new, if we were to draw another set on here, so if these replicate again, So as you continue replicating with the dispersive, you're always getting a random mix of old and new. And so you'd always be getting N14 and N15 mixed. So Meselson and Stahl were able to cross out the dispersive model, which left the conservative model. Or the semi-conservative model. So now if we go back and we finish our drawing here, on the right, we can actually finish drawing what actually happened to the DNA. So this strand of DNA split in two. We were then able to, in the N14, the other half was N14. So this is where half old, half new, half N15, half N14 comes from. In the next transfer, those were also split again, but because we, because we are in N14 media, we could only add N14 nitrogenous bases. So now we have two pieces of DNA where one is half N15, half N14, and we have two pieces of DNA where they're both strands are N14. And so again, showing us where those two bands came from. And we could actually continue this for another round of replication. If we had another round of replication, we would have the black and the pink split, the two pink split, and then we would get, since we're still growing in N14, more pink. So if we were to look at the cesium chloride gradient tube, we would still see two bands, but we would see that top band getting larger. And in fact, when Meselson and Stahl continued this, that is exactly what they saw. So in conclusion, what Meselson and Stahl showed us with their data is that DNA is replicated by the semi-conservative model. So now that we have figured out that DNA is replicated by the semi-conservative model, now we are ready for DNA replication at the molecular level. So this is going to be the end of video lecture 2, and in video lecture 3, when we come back, we are actually going to jump into, at the molecular level, how is DNA replicated. So thanks for hanging out with me, and I will see you in part 3. Bye everyone.